So in our previous lectures of genetic engineering, we have discussed about what is genetic engineering, what is the main aspect of genetic engineering, which is nothing but the cloning. So there are two methods by which we can perform cloning. First is with the help of cell and second is without the help of cell. If we perform gene cloning with the help of cell, it is known as gene cloning, and if we perform cloning without the help of cell, it is known as polymerase chain reaction. Then we have taken an overlook of gene cloning and polymerase chain reaction. Then we have discussed about importance of gene cloning and polymerase chain reaction. So in this video of genetic engineering, we are going to discuss about tools or components of gene cloning which are required to perform it. So there are four major tools which are required to perform gene cloning. The first tool which is required is gene of interest or a DNA fragment. So we have to isolate whole genome from an organism, and then we have, so consider this as a whole genome of an organism X, and we have to study about a specific gene known as AB gene, and we have to make numerous copies of it or clones of it. For that we have to isolate this DNA fragment from an organism. After isolation of this DNA fragment from an organism, then we ligate this DNA fragment into an vector. Here in red you can observe that the isolated DNA fragment will ligate it into the vector. So what are vectors? Vectors are nothing but vehicles which carries the DNA fragment into an host cell. Here when we ligate the DNA fragment into the vector, it is known as recombinant DNA molecule. After the formation of recombinant DNA molecule, we insert this recombinant DNA molecule into an host cell. Now you have clear that why we have that first step is that we isolated DNA fragment, then we ligate the DNA fragment into an vector. This ligation of the DNA fragment into a vector is nothing but known as recombinant DNA molecule. Then the third component which is required is enzymes. Here we have isolated and recombinant as we have isolated a DNA fragment, then we ligated this DNA fragment into an vector, and this is known as recombinant DNA molecule. For this ligation we require enzymes. To isolate this DNA fragment from an organism, we require enzymes. So enzymes also play a vital role to perform gene cloning. So enzymes are also the tools which are major tools which are required for performing gene cloning. So the fourth tool which is required to perform gene cloning is nothing but the host cell. Host cell can be any living cell. Here we have considered an example of an bacterial host cell. Consider a recombinant DNA molecule, and then we insert this recombinant DNA molecule in a host cell to make numerous copies or clones of it. So these are the four major tools which are required for gene cloning. First is an DNA fragment, then we ligate this DNA fragment into the vector and then the third major tool is required is enzymes, and the fourth major tool which is required to perform gene cloning is host cell. You must be wondering why we are doing such procedure just to make clones of a gene. No, the major procedure to make clones of it is right, but we also want to study gene structure, composition of gene, control of gene expression. For that also we are doing gene cloning. I hope you have understood what are the major tools of gene cloning which are required, what is the main purpose of gene cloning, why we are performing gene cloning. In a further video lectures, we are going to discuss in detail these four tools one by one. How to isolate DNA fragment from an organism, what are different types of vectors, what are different types of enzymes which are required, and different types of host cells or living things which are required to perform gene cloning. Thank you.